These are the lecture notes for Chapter 10 on cell reproduction. Okay. First, let's discuss the functions of cell division, the reasons that cells divide. Multicellular organisms use cell division for growth. They use cell division for maintenance and repair of cells and tissues. And single-celled organisms use cell division to reproduce. So when a multicellular organism is first created, it's created by fertilization of a sperm and an egg, of course, and that first cell that results is called a zygote. And that zygote, that original cell, in humans, that zygote is going to divide many, many times until it forms about, on average, 10 trillion cells. There's about 10 trillion cells that form the human body. So all of those cell divisions are the cells are dividing so that we can get larger. So growth is one reason for cell division, and then cell repair. So if we have damage to our skin, maybe we have a burn or we have some kind of wound or some kind of infection in our tissues, the cells that are lost and damaged have to be replaced, and those are replaced also by cell division. Cells in our body called stem cells. Stem cells can be embryonic stem cells, which can divide and produce just any general type of cell, or it can be a stem cell that only produces like red blood cells, or maybe it's a stem cell that only replaces skin cells when we shed our skin or when our skin is damaged. But those stem cells can divide and produce new cells to repair the cells that are lost or damaged. In a cell, the copy of the DNA, all of the DNA, all of the genes that the cell has, is called its genome. Now, in prokaryotes, the genome is different. Prokaryotes, remember, are they're either the domain bacteria or the domain archaea. These are cells that do not have a nucleus. So the genome is one double-stranded circular DNA molecule. It's a circular chromosome is what it is. Our chromosomes, we have 23 pairs of chromosomes in humans, and they are rod-shaped, but there's one single circular chromosome in a prokaryote. And here you can see it. It doesn't look much like a circle, but that is because it is so large and it's just tangled up. But it's really a very large circular chromosome, and it resides in a region called the nucleoid region. Sometimes you'll have additional loops of DNA called plasmids, but they'll be very small, and plasmids might contain extra genes. This is a microscopic photograph of what looks like onion cells. In fact, it looks like the very same onion cells that you have in your mitosis lab, which is lab 11. So the cells in the tip of an onion root are the ones that are dividing, because that's a region of growth in the onion. So what you're seeing here are cells in various stages of cell division. In fact, while I've got this picture, I'll just show you what the different stages look like. When we go through cell division, we're going to talk about mitosis first, and there are 4 stages: prophase, metaphase, anaphase, and telophase. And then there is also the stage that comes before mitosis called interphase. So I wrote this mnemonic device IPMAT, IPMAT. If you remember IPMAT, that is the order of the stages. So a cell that's in interphase would look like this. You see how the nucleus, inside the nucleus, you can't really see the individual chromosomes because the DNA is scattered out. It's spread out and scattered, and in fact, the DNA is called chromatin in this stage. So this is what a nucleus looks like of a cell that is in interphase. And you see most of the cells on this image are in interphase. There's a whole bunch of them. Here's one, here's one, here's one. These are all in interphase. When the chromosomes start condensing, the DNA is no longer called chromatin, it's called chromosomes. When you start to see them condensing, but they're still scattered, that is prophase. There are several cells that I see that are in prophase. Here's another one. Here's another one. And then the next stage is metaphase. The chromosomes are going to line up in the middle of the cell. And this is a cell that's in metaphase. They don't form a perfect line, but they do line up in the middle, like as if there were an equator or an imaginary line going down the middle of the cell. The chromosomes line up at that metaphase plate is what it's called. And then I do not see an anaphase, but anaphase is when those chromosomes begin pulling apart. And then telophase, you're going to have in a plant cell, you're going to have a new cell wall begins forming as a cell plate. I do not see a very good example here of anaphase or telophase, but at least you can see interphase, prophase, and metaphase. So the eukaryotic genome consists of chromosomes. Eukaryotic cells, remember, are cells that have a nucleus, and the domain eukarya consists of fungi, protista, plants, and animals. And we know that humans have 46 total chromosomes, and other organisms have different numbers of chromosomes. The number of DNA molecules or chromosomes varies among species, as I just said, and within a species, the chromosome number is very consistent. So in other words, unless a human has a chromosomal abnormality, they have 46 total chromosomes. And you can see here, I'm pretty sure HC stands for human chromosome. So there are 23 total pairs of human chromosomes. And what you can see in these 23 pairs is that this is a male, because instead of having two X chromosomes at pair 23, this individual has an X and a Y chromosome. And so that's a male. If it were two X's, it would be a female. Okay, so cells in the body that are not sex cells. So if they're not sperm or egg, we call them somatic cells, just body cells. These cells can be nerve cells or skin cells or blood cells, bone cells, any of the cells of your body that are not a sperm or an egg. They are called somatic cells. And somatic cells typically have two sets of chromosomes, and so we call them diploid. In humans, the diploid or 2N number of chromosomes is 46, or 23 pairs. In gametes, the gametes are the eggs in females and the sperm in males. They're going to have half the number of chromosomes, so we call them haploid, or 1N. And the haploid number for humans is 23. So in a human egg or a human sperm, there are going to be just 23 chromosomes, because the mom only gives half of her chromosomes and the dad only gives half of his chromosomes to form the offspring. Now, an image, a picture of chromosomes that are arranged by size and pair is called a karyotype. We just looked at one. It's a karyotype when you arrange the chromosomes in pairs, line them up, starting with the tallest and then down to pair number 23, which are the sex chromosomes, whether they're two X's or an X and a Y. So chromosome pairs are called homologs. And the chromosome pairs in diploid cells are called homologous because they're homologous pairs because they have genes for the same traits. Now, that does not mean one of the chromosomes in each pair comes from the mom and one comes from the dad. So that does not mean that they have the same traits. But for example, they might have the gene for eye color or the gene for whether or not they have freckles, but they would have the same genes. The homologous chromosomes would have genes for the same traits. They just might not be the same version of that gene. So they're homologous when they're the same size and they have genes for the same traits. Now, the only pairs that don't match in humans are the X and the Y chromosomes if you are a male. Now, the two X chromosomes do match up. They're the X sex chromosomes. If you are a female, the two X sex chromosomes on pair 23 are the same exact size. But if you're a male, you have an X and a Y chromosome on pair 23. So we would call them not homologous, but heterologous because they're different. This is an image of DNA in the various stages so that you can understand the difference between naked DNA versus a chromatin versus a chromosome. So DNA, just DNA, is the double helix structure. And what happens when DNA forms into chromatin inside of the nucleus of our cells is that first the DNA wraps around proteins called histones. So you can see the DNA is wrapped around a histone. The histone is the various colored protein that's inside here. Definitely got a quaternary structure, quaternary level of structure. And then that structure is called a nucleosome, and the nucleosomes coil into chromatin. And chromatin can condense, and when chromatin condenses, it is called a chromosome. So chromatin is DNA coiled around histone proteins that is spread out. And it's spread out to the point that you really can't make out any shapes when you're looking at the cell in the microscope. But when it's time for the cell to divide, the chromatin condenses and forms chromosomes, which have very obvious rod-shaped structures. In fact, when they're attached, they look more like an X than a rod. They're actually two rod-shaped structures attached in the center. But that is what the DNA, that's the form the DNA takes whenever the cell is about to divide, is it condenses and forms chromosomes. So these notes just explain what I just explained, so I'm going to skip through them. The packaging of DNA into chromatin and then chromosomes. And this video is on slide 10, so if you would like to watch it, I encourage you to watch it. You can go to slide 10. Now, when we come to our next part of the lecture, part 2, we're going to discuss the cell cycle, which is the series of events in the life of a cell from the time that it's formed from the time it's produced until the time that it divides again. And so these are the stages. You have the mitosis and cytokinesis, and then you have interphase, which consists of G1, S, and G2 stages.